Good morning everyone. My name is Faqihah binti Mama Azlan. So today I will be presenting a research article entitled Deep Eutectic Solvent Based Ultrasound Assisted Extraction of Flavonoid from Houttuynia cordata by Wu and colleagues, published in the journal Food in 2025. So, flavonoids are important plant bioactive compounds known for their antioxidant, anti-cancer, anti-inflammatory and cardiovascular protective properties. However, conventional extraction methods often rely on organic solvents such as ethanol and methanol, which can be toxic, volatile, and environmentally unfriendly. Therefore, the researcher aimed to develop a greener and more efficient extraction method by combining deep eutectic solvent or DESS with ultrasound-assisted extraction. The knowledge gap addressed in this study is the limited information on the use of DES for extracting flavonoids from Houttuynia cordata, a medicinal plant widely used in Asia. The hypothesis was that DES-based ultrasound extraction could improve flavonoid yield while maintaining biological activity and reducing environmental impact. This study began by preparing 10 different choline chloride-based deep eutectic solvents using various hydrogen bond donors such as lactic acid, glycerol, and urea. So, the researcher first screened this solvent to identify the most effective one for flavonoid extraction. Next, single-factor experiments were performed to evaluate the effects of extraction parameters, including extraction time, temperature, water content, ultrasound power, solid to liquid ratio, and solvent molar ratio. After determining the important factors, response surface methodology or RSM using a box blanket design were employed to optimize the extraction condition. Finally, the extracted flavonoid were analyzed to antioxidant activity using DPPH, ABTS, and hydroxyl radical scavenging assay, as well as nitrate scavenging activity. One limitation mentioned in this study, which is the high viscosity of DES. It can make a downstream separation and purification more difficult. The first important finding was the identification of the most effective solvent. So among all the 10 DES tested, the combination of choline chloride and lactic acid, known as CHClLa, produced the highest flavonoid yield of 66.31 mg per gram. This extraction efficiency was significantly higher than the conventional 16% ethanol extraction, which only achieved 54.61 mg per gram. So this result demonstrated that DES can outperform traditional organic solvent while providing a greener extraction approach. The next significant finding was the strong antioxidant activity of the extracted flavonoid. At the concentration of 1 mg per milliliter, the DES extracted showed from the graph. So 93.10% DPPH scavenging activity, 99.18 ABTS scavenging activity, and 40.60% hydroxyl radical scavenging activity. These values were higher than those obtained using ethanol extraction. So this suggested that the DES extraction method not only increases yield but also preserves the biological activity of the flavonoids. After optimization using response surface methodology or RSM, the ideal extraction conditions were identified as 60 minutes extraction time, 40 degrees Celsius, ultrasound power of 420 watts. So under all these conditions, the flavonoid yield reached 79.86 mg per gram. So the predicted value from the model was 81.42 mg per gram, which was very close to the experimental value. This indicates that the model was highly accurate and reliable. The author interpreted this finding as evidence that combination of DES and ultrasound creates a highly efficient extraction system. Ultrasound generates cavitation effect that break plant cell wall, while DES enhance flavonoid solubility through hydrogen bonding interaction. From a theoretical perspective, this study advanced the field of green extraction technology by demonstrating how environmentally friendly solvent can replace conventional organic solvent without sacrificing efficiency. So practically, this method could be applied in food, nutraceutical, pharmaceutical, and cosmetic industry to produce natural antioxidants more sustainably. The extracted flavonoid exhibits strong antioxidant and nitrate scavenging activity, suggesting potential application in functional food and health products. For future, for future research, the author suggested that developing faster and more efficient method for separating target compound from DES, since their high viscosity remain a challenge. In conclusion, this study successfully developed a green and efficient method for extracting flavonoid from Houttuynia cordata using deep eutectic solvents combined with ultrasound-assisted extraction. The optimized process achieved a high flavonoid yield of 79.86 mg per gram and produced extract with strong antioxidant and nitrate scavenging activity. This research highlights that the potential of DES as a sustainable alternative to conventional organic solvent and contributes to the advancement of green biotechnology. To end off my presentation, I would like to leave you with a question. If deep eutectic solvents are more environmentally friendly and equally effective, should they replace traditional organic solvent in large-scale industrial extraction process? Thank you for your attention.